Bismillahirrahmanirrahim. This is the final revision video for the MCQ, and I hope I can handle some of the questions which you all have been asking me on WhatsApp and in your comments. So I hope this is going to be a helpful video for you all to watch just before your exam. First, let's revise the structure of DNA and what is DNA. So a DNA, this would be one chromosome. So a DNA this is now one chromosome is made up of one molecule of DNA. But one molecule of DNA contains two strands. This is one strand. This is the second strand. So one strand and two strands. So two strands is one molecule of DNA. So a chromosome is made up of one molecule of DNA. Now here what do you see? You see pairs of chromosome in a human cell. So there are 23 pairs of chromosomes. But actually, how many are there? 46 chromosomes. So 46 chromosomes, but here we are seeing 92 chromatids because each chromosome copy has been formed. So this is the copy of it which has been formed. So there will be how many? So there will be 184 DNA molecules because one chromosome is one DNA molecule, and then when a copy of it is made, so this will be two DNA molecules. So two chromatids, two DNA molecules. So everybody has to be very clear on this. Of course, we don't study meiosis in AS level, so I won't talk about that. So one molecule of DNA is made up of two strands of DNA. Then what is a gene? Gene is a portion of DNA which codes for a protein or a nucleic acid. Please remember, even the nucleic acid like RNA, mRNA, tRNA, and rRNA are also coded by a gene. So gene also code for proteins and also code for nucleic acids. Then another thing, we must know the 5-3 direction. So 5-3, this is 5-3 here. This is because the 5 carbon is at the top. This is the 5 carbon. And here, because we have the 3 carbon here. So this is what we have to understand is why do we call it the 5 carbon? So this is the 5-3 direction in which the DNA polymerase works. And this will work in this direction, and this is called anti-parallel. Now, something which you all have to remember, adenine and guanine are purines. And purine has a U in it, so there's a double ring structure. You can see this is a double ring structure. This is also a double ring structure. So purines double ring. And there's a U here, there's a U here, there's a U here, and there's a U here. So double ring structure. Now the other three, cytosine, thymine, and uracil, these are pyrimidines. These are pyrimidines and they are single ring structure. You have to ratify it. You have to memorize this. So cytosine, thymine, and uracil are pyrimidines, while the purines are adenine and guanine. So I tell you to remember it like this: adenine and guanine. So this is the month of August. So AUG. Then the base and the ribose or the deoxyribose, this is called nucleoside plus phosphate, gives you a nucleotide. So a nucleotide is made up of a deoxyribose or a ribose and a phosphate and a base attached to it. Then the difference, deoxyribose. Now you can see here, this is carbon 1, this is carbon 2, this is carbon 3, this is carbon 4, and this is carbon 5. This is not 6, please remember. This is not 5, the one, oh, there's an oxygen bridge here. So on carbon 2, you have hydrogen, so it's deoxy. And here there is OH, and this is ribose. So this is something you've got to remember. Deoxy means less oxygen. So there's an oxygen less here, there's an OH here in RNA. So RNA has ribose and DNA has deoxyribose. They're both pentose sugars. They're both pentose sugars. Then the bases are, of course, we have thymine here. So in DNA, we have adenine, thymine, guanine, and cytosine. In RNA, we have adenine and uracil. So there is no thymine. But A, G, and C is common to both. But here is the only difference is the thymine and the uracil. So whenever you see a T, it has to be DNA, it can't be an RNA. And just now somebody asked me a question on WhatsApp, and I was looking at the person how he was getting it all wrong, that he was looking at thymine and saying this is an RNA molecule. Now you can see the difference between a prokaryote and a eukaryote. Now you can see in a prokaryote, you must remember the gene must be in part of the circular DNA. So this could be one gene, this could be another gene, and this would be part of the circular DNA. And then, of course, transcription takes place, and then the mRNA gets attached to the ribosome and the protein is made. While here in a eukaryote, you can see there's a difference. Is the nucleus, there's going to be part of a chromosome, and this is going to be copied by the pre-mRNA, and then the pre-mRNA is going to be formed the mature RNA or the mRNA, and then it's going to go the translation. So just a brief recap of how this is different in both prokaryotes and eukaryotes. Another very pleasant diagram showing you this. Why I show you these diagrams is because you form a mental image of these processes, and then it helps you in the exam to actually rethink these answers when you're doing the MCQs. This is again the new syllabus, how alternative splicing mechanisms can result in different primary structure resulting in two different proteins from one enzyme. And you know the exons would remain and the introns would be removed, but then the exons would be put together in different order. And this would of course give you protein 1 and protein 2. What we need to understand is this is a very good diagram of transcription. The transcription, you can say we need an enzyme called RNA polymerase, and that is going to result in a certain section of DNA being copied by the RNA, and we get an RNA transcript. And then number one, RNA is transcribed from a DNA template. This is a word used, DNA template. Then in eukaryotes, the RNA transcript, the pre-mRNA is spliced and modified. What does it mean spliced? These introns are removed. So this intron is removed and this intron is removed. Now all what do we have is we have all the exons attached together, and now this exon, now this is called the mature RNA or the mRNA, and now this will leave the nuclear pore and will enter into the cytoplasm where it will attach to the ribosome. So here the ribosome is attached. Then we have amino acid activation. What is amino acid activation is that the tRNAs must get attached to some appropriate amino acid. So there's an enzyme called amino acyl tRNA synthetase, which actually attaches the amino acid to the appropriate tRNA molecule and attaches it. And this is called amino acid activation. So this is an activated amino acid. Now this will bring it, and here you can see the anticodon and the codon is matching it. And here we see the appropriate amino acid being brought, and then the amino acids are going to be linked. So each amino acid then attaches to the next amino acid with the help of a peptide bond. So this is the whole process of transcription and translation. Now I want you to understand this because I can think of a possible question which can come on this. When we say it's a phosphodiester bond, that means two ester bonds. So there's the first and the second ester linkage. So now if I ask you in a DNA molecule, there are 6 nucleotides. So how many ester bonds will there be? I haven't said phosphodiester, I've said ester bonds. How many will there be? So this is now what you've got to figure out, how you're going to answer this question. Now the common mistake that you make is when you look at this diagram, what are they? There are 8 nucleotides. 1, 2, 3, 4, 5, 6, 7, 8. But there are 4 base pairs. So how many phosphodiester bonds would there be? And how many ester bonds would there be? So phosphodiester bonds would be 1, 2, 3, 4, 5, 6. So 6 phosphodiester bond, but ester bonds would be 12 because it's a diester, disaccharide, dipeptide. Dipeptide means 2 amino acid. Disaccharide means 2 glucose. Maltose is a disaccharide made up of 2 glucose molecules joined together. So so it will be 6 phosphodiester bonds but 12 ester bonds. And I can think of a possible MCQ on this. Of course, I've seen some in some papers, but this would be a new MCQ. Another question which is often asked is the minimum number of hydrogen bonds. What does it mean by minimum number of hydrogen bonds? Now you've got to understand minimum number of hydrogen bonds means if they were all were 80. So this is 80. What if this was 80 as well? What was if this was 80 as well? This is of course 80 as well. So there would be minimum number, you know, A and T has only 2 hydrogen bonds. So these would be 8 hydrogen bonds. But if I say maximum number of hydrogen bonds, then what do I expect you to say? What I expect you to understand is that we're expecting you to say that all of these are C and G. So G and C here, C and G here, then here also C and G, and here also C and G, or G and C other way. So this would be 3. So 1, 2, 3, 4, 6, 7, 8, 9, 10, 11, 12. So maximum number would be 12 hydrogen bonds. This is what they expect you to answer when we ask you minimum number or maximum number of hydrogen bonds. Now in this question, you often ask me this question, what is the minimum number of hydrogen bond in the length of DNA containing 700 nucleotides? That means, what does mean 700 nucleotides? It means there is one A here, there's T here. So this is two nucleotides. So there is T here and there's A here. So another nucleotide. Then C here and G here. So you here, what do we have? We have 6 nucleotides. But 3 on one side, 3 on the other side. So in this situation, what would we have? We would have 350 on this side and 350 on this side. So if I said there's 6 nucleotides, so I mean 3 on this side and 3 on this side. So if I asked you 700 nucleotides, so it would be 350 on this side, 350 on this side. Minimum number means 2. It means all of them are A and T. So 2 hydrogen bonds. So 350 into 2 is 700. This is how we calculate the minimum number of hydrogen bonds. Another question which is often asked is, what is the minimum layer of phospholipids that a molecule of oxygen passes? Now why is the answer 10? Because each phospholipid, each cell membrane is made up of 2 phospholipid molecules. It crosses 5 cell membranes. I've done this in a previous video as well. It crosses 5 cell membranes, so 5 into 2 would be 10 because each phospholipid molecule is one cell membrane. What are telomeres? What are telomeres? Telomeres are the chromosomes, the ends of the chromosomes, right? This is a telomere, right? This is a telomere. Now, how much are these telomeres made up of? They are made up of anything from 8,000 to 10,000 nucleotides. You can Google it. I said it's made of 10,000, 8,000 to 10,000 nucleotides. As mitotic division starts and more mitotic division occurs, these become shorter. When they become shorter, that means the number of nucleotides will decrease. Maybe they've gone down to now 6,000, or they've gone down to 4,000, or they've gone down to 3,500. So in all this situation, what was there? The smallest number. You have to look at the smallest number: 5,700, 5,645, 5,580, 548, 5,548. So the smallest number would have shown the most mitotic divisions. So the most mitotic division means more and more of the telomere is decreasing. The number of nucleotides is decreasing. So which total number of bases was found in the cell that had undergone, and total number of bases in the telomere were determined. So what is the total number of bases found in the cell that had undergone the most mitotic divisions? A student asked me this question and said, why isn't the answer C? And you know, it really got me very angry because I get angry very easily because Pathan DNA. So I said, how can it be C? And he said, Miss, why can't it be C? And I said, well, the reason is mRNA transcribed from the HBS allele will contain the codon CAC. Well, that's of course you figured that out. Instead of the codon CTC, this was the catch in it. How can the mRNA have a T in it? I've told you this million times, there can't be a T in the mRNA. And when the student asked me this is, why can't it be C? I mean, why did I get angry? I got angry for this very simple reason, is because you're not reading the question. And if you're going to make these silly mistakes, that you're going to say CTC, but miss, then CTC is right. Yes, because you had figured this out, this was CTC here. But GAG, you got this wrong. The mRNA cannot have CTC in it. So you've got this wrong here. Your rough work has been incorrect. So please understand this is that we cannot have any T in the mRNA. So whenever you do this, please do the rough work very carefully on the question. You can do rough work on the question and then figure out which is the correct answer. If you're struggling with it, then leave it, because now you will have 1 hour 15 minutes. So you can come to the difficult questions again and then you can redo them and think about them and do it a little more with a lot of thought in it. So, okay, which one is right or which one is wrong? And then think about each one which is wrong and then mark the one which is the correct. You must take your time on these difficult questions. Now I would like you to pause this here and read this very carefully and then stop the video here, pause it, and then tell me the answers. How many amino acids does this gene for an enzyme code for? The total number of base pairs in this gene are 150 base pairs Y. Now if you add 12, 27 and 60, that is 99 base pairs, and you divide that by 3, so it codes for 33 amino acids. It codes for 33 amino acids because 3 bases code for 1 amino acid. So 99 divided by 3 is 33 amino acid it codes for. So it codes for 33 amino acids. But if you add these are 99 base pairs, but I'm saying the gene is 150 base pairs. Why? Why? Because we didn't count the introns in it. Now I can expect such an MCQ coming because they are going to ask you, well, why is it not, you're saying 33 amino acid, but they're 150 base pairs. So this is one way of looking into these questions which you can be asked, and these are very challenging questions because now remember your time has been increased, so you are going to be asked some challenging questions. Now the same question I can word it in a different manner. The same question I've shown you how I can word it. What do I mean by 12 base pairs? It means AT is one base pair, AT. So 12 base pairs. This is the diagram which I've shown you. So I can say here 12 base pairs, or I can say 24 nucleotides. So if you say there are 12 on this side and there are 12 on this side, so there are 24 nucleotides. So please get this right is how we word it. You must read it very carefully. Are we talking of base pairs or are we talking of nucleotides? So this would be 24 nucleotides, which I've shown you here. But it can also be worded as 12 base pairs. So the same question I can show you in different colors. I've shown you how I worded it. So the 12 base pair would be 24, the 27 would be 27 plus 27 would be 54, and 60 plus 60 would be 120 nucleotides. Now, in the new syllabus, we have to learn the leading strand and the lagging strand. Now, what you have to understand is that the DNA polymerase can only work in the 5'3" direction. So this will keep on going and keep on going and keep on going and keep on going. But you see, a portion of DNA unzips. So this portion of DNA has unzipped. So we're getting here, this portion of DNA is unzipped. Only this much portion is unzipped. But then the further as it unzips, now we have a problem. Because you see, this has to be copied discontinuously. So we have these Okazaki fragments. And the Okazaki fragments are only in the lagging strand. And then, of course, we need an enzyme to join these. And we have these RNA primers, and the RNA primers are then replaced by the DNA. So what you have to understand is that leading strand. Now, for the leading strand, this is the 5. This is the 5 and this is the 3. So this will be right like this. But here now, this is the 5. So the 3 has to come here. So this has to be done in this direction. But this direction cannot be continuous because as this unzips, then this has to continue. So this one has to open up. So you've got to understand is this DNA helical, the DNA double helix has to keep on opening a little and little, and there is this continuous DNA replication has to occur. So this is something which you must understand is that the Okazaki fragments, there's the lagging strand is discontinuous and the leading strand is continuous. Now you must be wondering why have I done that? Well, I've done that for a certain reason is that there is one way of doing this. When you get this wrong, if you get this wrong very frequently, then you can do from DNA to mRNA, you can do this. So this is actually just a DNA because you've got the T's here. So for every A, you have a T here. So now, but if you are struggling with this and you get this wrong many times, then you need to now assess this and realize, okay, first do the T's, but then convert these into U's. You can do this as well because some of you get confused and you don't do the U's and sometimes you do a T as well. Now, of course, you can do the tRNA. So the tRNA U was a T, so it is an A here and the A here, then this was an A, so then this can't be a T here. This has to be a U here. Or you could have written a T and then converted into a U. U would be an A. G and C is of course fine. You don't make a mistake. Is then U would have to be, if it's a T, so there should be an A here. Then these are A's here. So A would have not, you can't have T here. But if you have written T, then you have to write a U on top of it. and then you have to correct yourself. So maybe if you want to do it like this, I mean, it's up to you. You can do a correction later on of the U if you feel that is the way you want to do it. But I'm just giving you these ideas how to go about it, is that you have to figure it out. That now when you look at the DNA and the tRNA, you look what is different. Everything except is the same except the T's have become U's. So the T's have become U's. So that's a very interesting phenomena that you can see if you want to do it like this. I'm just giving you these choices because you tend to make these mistakes. Maybe you're nervous, maybe you in the exam you know it all, but sometimes you do it by mistake. It's a careless thing, or maybe you just do it. So this is one way of handling these questions. A little bit about the amino acid activation. The main thing which I want you to remember is it requires ATP. So energy is needed, ATP is needed, or energy is released. So aerobic respiration will result in ATP formation, and then ATP is needed for the activation. So the methionine, which is an amino acid, has to attach to a tRNA, which is specific for methionine. So you can see this enzyme, amino acid tRNA synthetase, is specific for this amino acid. And it will join the amino acid to the tRNA molecules. Now the ribosomal structure, I want you to know about the three sites which are called the EPA. So nine bases can fit in here because three bases code for one amino acid. So three here, three here, three here, so nine. And this is called the exit site and the peptidyl tRNA binding site and the amino acid tRNA binding site. So this is the mRNA and this is the large subunit and the small subunit. Now the A site binds to the tRNA anticodon that carries the proper amino acid. P site binds to the last entering tRNA that holds the growing polypeptide chain. And the E site allows the exit of the empty tRNA. So I just wanted to know this is sequence, and this is how it is going to be done in the translation process. Now another thing about the RNA, this is the transfer RNA, the tRNA. So this is the amino acid attachment site. But the main thing which I want you to see is there are hydrogen bonds between the paired bases. The reason being is that it is a single strand, but it is folded on itself. So, I mean, you usually think, okay, mRNA is just a single strand, and tRNA is also a single strand, and rRNA is also a single strand. But please remember, in tRNA, there are hydrogen bonds between the paired bases because this folds upon itself. And that is why this is the area where we've got these hydrogen bonds here, and then we've got these hydrogen bonds here. But some places are looped and these do not have any hydrogen bonds. Like this area, there are no hydrogen bonds. This area, no hydrogen bonds. This is a loop, there's no hydrogen bonds here, there are no hydrogen bonds here. And of course, this area, the three bases, these are called the anticodon. And you can see these in these two diagrams. And the reason why I said this is because I could think of an MCQ which could be checking this, whether hydrogen bonds are present in tRNA molecules or not. Yes, but not in mRNA, not in ribosomal RNA, but in tRNA, yes, hydrogen bonds, because the same single strand molecule folds upon itself and gives this clover leaf pattern. And this is why I wanted to give you this. That finishes this video, and thank you very much once again. Thank you everybody for watching these videos, and thank you for subscribing, and I hope you do very well in the paper one. And inshallah, we all expect a very brilliant result on the 11th of August, and all the best, and all my prayers with you. Thank you for watching my YouTube channel. Thank you for the positive comments. Thank you all the students who have asked questions so I could improve my teaching skills. Thank you once again.